Okay, another analytical biochemistry lecture. Although some of you may be coming from the molecular biology subject, and if you are, welcome. Some of this will be of interest to you, but this is mainly a more technically focused lecture about how proteomics can work. So, without further ado, first thing I'm going to do is I'm going to give you a little bit of background. Um, so hopefully most of you will understand the difference between genotype and phenotype. But if this concept has never been pointed out to you, here's what happens. So most of you know what the genome is. It's the DNA content of every cell in your body. And if you look at the genome from different cells, such as in this diagram where we've got bone cells and we've got lung cells and brain cells, the genome in all of these cells should essentially be the same. The genome is the blueprint of what could be made. They're the set of instructions of what could be in your body. Now, it's quite obvious if you look at yourself or if you look at this diagram that all of these different cells are different. They all contain the same genome, but they are phenotypically different. And what the phenotype is what is actually being made. So that is somewhat dictated by the genome, but it's more dictated by messenger RNA expression, by non-coding RNAs, by the proteins and proteoforms that are going to be expressed, which is going to be the main focus of this video, and also the lipids that are present and the metabolites that are present. Now, these vary from cell to cell. And so, yes, cells are different. So different cells are different because they express different genes and therefore they make different proteins. So that's the difference between genotype and phenotype. Just to hammer this home a little bit more, I love my Lego analogy here, where the box of Lego and the instructions that come with it can be considered to be the genome. But you've essentially got a box of parts that you can make a number of different kits out of, or a number of different cars and boats and things like that. These are actually the mature phenotype. So you've got the same set of basic parts, but they're just arranged in different ways. And this is showing you how the different cells are. Now, genes are often expressed because of feedback that happens. So the genotype in this looks like it's a very linear diagram where the genotype becomes the transcript and it leads all the way through to the phenotype. But this is actually incomplete because what really happens is that the microbiome, the metabolome, the proteome, the transcript, all of these things actually feed back on the genome to change what is being expressed. Now, one thing that people should be aware of is that just by having more genes doesn't mean that something is more complex. So this is the size of the genomes of a number of different organisms. So E. coli has about 4,500 genes, which is different to something like Staphylococcus that has 2,600. Yeast, the smallest eukaryote, that has about 6,500 genes. But then you get up to higher to animals where you're up to about 23,000 in a chicken, but a plant has more genes than a chicken does. So would you argue which one of these was more complex than the other? To take this to the logical extreme, and when this slide was first made, George Bush was in power and had just announced the victory in Iraq. And back then, we found out that there was about 25,000 genes. That's actually been revised down, and I've updated this slide for more modern times. It's not 25,000 anymore, it's more like 19,500, and this number keeps getting reduced down. So, what this implies is that genome size doesn't increase the complexity of the organism. So, only 1.5% of the human genome actually encodes for proteins. So you can see that, yes, up to a point, genome size does give more complexity, but there's not that much difference between us and dogs, and there's virtually no difference between us and other primates. So it's more important not how big your genome is, but when and how it is expressed. So, to put this into perspective, a few years ago, we started to get these drafts of the human proteome, and in this particular case, in this particular publication, they did 30 different cell types. And what they found was that this is a very important table that's actually buried in the supplementary data. And what it shows is that there are 2,350 genes that were expressed in all 30 cell types. So these are the housekeeping proteins. These are the proteins that need to be there for every single cell. So for things like ribosomes and other translation machinery. But then at the other end of the spectrum, you've got 1,500 genes that are only found in a single cell type, or 1,000 or so that are only found in 2 cell types. It's down here. These are the genes that are unique to a certain phenotype of a certain cell. These are the things that make certain cells unique. On top of that, again, just to ram the point home, where we find that 73% of proteins are found within all cells in this particular analysis, but the abundance of the particular proteoforms differs between each cell type. So here you've got a range of different, in this case, cancer cell lines, and each one of the bars that runs across here is the same proteoform. And what you can start to see is the different colors are the expression level, the abundance level of these particular proteins. When we talk about proteins, we talk about abundance, not gene expression. We're talking about the amount of a particular protein that occurs in a cell. And so what you can see here is that going from blue to red, each cell type has quite a significant difference in the abundance of a particular protein. This is reinforced by this diagram down here, which is showing you the correlation, the complete correlation of the genes being expressed and the genes being expressed and the abundance of those proteoforms across different cell types. And you can see that some of them have quite a high correlation, but some of them have a low correlation, but none of those correlations is 1-to-1. It's all quite different. Extending from this, a lot of people don't necessarily realize that there is very little correlation between the amount of messenger RNA and the amount of protein that is in a particular cell. So a lot of scientists at one stage believed that you could just use the transcriptome to get phenotypic information about what is going on in the cell. And when you actually look at the correlation between the amount of a particular RNA and the amount of its corresponding protein, there is very, very little correlation. So you can't use transcriptomics alone to predict what the phenotype of a cell is going to be. And in saying that, you can't use proteomics alone. We will come back to systems biology later on. And just one last graphical way of describing this. These two organisms have the same genome, but they are, after metamorphosis, they are phenotypically different because they have changed their proteome, changed what proteins, the abundance of different things that are being expressed, which give them a different phenotype. So what is the proteome? It's all of the proteins that are being expressed by a cell, an organism or tissue under a certain set of environmental conditions at a certain point in time. All of those things are very important because environment has an effect on the proteome. So genomics measures genotype. It's relatively straightforward to do, and high throughput next generation sequencing is relatively straightforward these days and can generate an enormous amount of data. Proteomics is a measure of the phenotype of the cell. The phenotype is also defined by the metabolites and the amount of different metabolites that are there, and lipids are also a form of metabolite. Those lipids and their abundance is a measure of phenotype. So a cell is not just what proteoforms are being expressed or what the abundance is. It's when is that protein being produced, how much of it is being produced, how is it being modified after it's being produced, the production of proteoforms. But it's also, what are those proteins interacting with? Are they interacting with other proteins? Are they interacting with the same proteins? Are they interacting with DNA, with metabolites, with RNA, with lipids? And this is called the interactome. The purpose of doing proteomics is to obtain this global, system-wide view of what's going on in a cell under a certain set of conditions. And what we want to do is we measure the difference in abundance of those proteins to see what's going on. Now, what we want to include is also the analysis of the modifications of these proteins, because the modifications that happen on proteins, whether they're cleaved into smaller functional proteoforms, whether they're phosphorylated, glycosylated, there's 350 odd different modifications that can be put onto proteins. None of these things are they're not predictable by the genome. So the challenges of proteomics is actually to quantify the changes in the abundance of proteins and proteoforms in the system. This is another graphic example trying to show you the complexity of what's going on, where the genome is the tip of the iceberg, but then we get transcripts and alternative processing and editing and slicing to create an estimated 100,000 transcripts from these 20,000 genes. But then from the transcriptome to the proteome, we get estimated 1 million different proteoforms in a particular cell. And these can vary with time and with the location of the cell and the location within the cell. You also have all of these modifications and interactions and movement affecting this. So the problem is that we want to be able to balance the amount of time and effort that it takes to extract and measure the proteins, be able to measure PTMs, be able to measure interactions, and therefore understand what the hell is going on. So why do we study the proteome? I've alluded to this a little bit. We do it to determine the phenotypic changes that occur in a system. We can use this to ascertain the function and interactions of proteins during disease or during infection. We can work out the mechanisms of cellular function and how they change with disease or infection. We can look at protein interactions and protein complexes and how they alter in these diseases. But most importantly, we want to find biomarkers and targets for either early therapeutic intervention or early diagnosis of disease. These 2 points are really the main focus of doing medical proteomics. So now I'm going to walk you through an example from this article that was done by some group at Sydney University. And what they were doing was they were looking at the effect of fasting on the proteins that are present in plasma. But it may be subtle. I mean, the color scheme here looks relatively similar, but there are some very distinct popping up differences, where you can see subtle changes in the abundance that's due to fasting, but you can also see some protein sequence variation patient to patient. And this goes to show the variation that occurs across people, but over the course of, the variation that happens within a population. The idea that everyone is the same is a fallacy. When we measure the proteomes of relatively healthy people under the same conditions, the abundance of particular proteoforms can be quite different. And this is the kind of output that you see in these cases, where this is a volcano plot, and the further in the top and the further away from the axes, the more the fold change of abundance is, and the more significant the changes are. So you can see things like this apolipoprotein that are being increased in expression due to fasting, and other ones that go down in abundance. So the other thing that you can use proteomics for is to start to see sequence variations between patients. And so this is looking at the same a variant peptide in apolipoprotein A4, where there is a serine to asparagine conversion, and you can measure how many people actually have this variant. So this guy here, when you look at, he has, or this person, who don't know if it's a male or a female, they have exclusively this peptide with the serine in it, whereas some people are actually converting between the two. So this person, when they're before they're fasting, they have a almost 2-to-1 ratio of these two variants, and during fasting it actually flips over and goes the other way, where this variant becomes more common than this variant. And what does this mean? Who knows? You've got this one here, which is apparently involved in diabetes complications, and you can see that fasting can flip these around in some patients. It's just showing the patient-to-patient variation. So these are the kind of things that you can use proteomics for. So this is one of the take-home messages from the paper, where we identified significant abundance differences induced by intermittent fasting, increased apolipoprotein A4, decreased C2 and C3. This then changes the amount of plasma triglycerides, and that is therefore changing metabolism. So we proposed that intermittent fasting had a positive effect on lipid metabolism through modulation of high-density lipoproteins. So this is now showing not only the proteome analysis, but the integration on the wider system. So I'm just going to start talking about some of the problems that we face when we do proteomics, because it is quite complicated. So one of the problems that we have is there is more proteins than genes. So this comes about through alternative splicing when we make transcripts, but it also comes to this idea of post-translational cleavage of proteoforms into smaller proteoforms, and further modification of those proteins by putting on glycosylation or phosphorylation. So this is an estimate where these 20,000 odd, and this should be 19,500 human genes, produce about 2 million proteoforms. The other problem with that is the issue of the difference in abundance of all of those proteoforms, but I'm going to come back to that in a few slides. The next problem is that every time we modify a proteoform, we create another proteoform. So we can have isoforms of the same gene creating different proteoforms, but then within a particular isoform, we can get glycosylation differences, we can get phosphorylation differences that are making different mature proteoforms. And this all has to be dealt with. There's 936 modifications in UniProt, but a lot of these are not well understood and are not well characterized. So this shows in the database, phosphorylation is relatively well characterized, but the others are poorly characterized. And this is because it's very difficult to isolate these modified proteoforms away from everything else. and they tend to be in lower abundance, and therefore they are more difficult to detect. Again, I'll come back to that in a couple of slides. DNA and RNA is relatively easy to work with. We've got this wonderful thing called the PCR reaction, where we can take a piece of DNA and we can amplify it from a small number to an enormous number of molecules and increase the sensitivity of the assay. So one of the reasons that genomics is so successful and we are able to sequence genomes relatively easily is because of this PCR reaction, where we can boost sensitivity just by making more molecules of the thing that we want to measure. In proteomics, we don't have that. So we've got this problem of dynamic range. So as I mentioned, genomics and transcriptomics, we can amplify our signal, but in proteins, we don't have the equivalent of PCR. We can't amplify the amount of particular proteins to boost sensitivity. So if we want to detect more of these low abundance proteins like the interleukins and the cytokines, the obvious idea is that we need to start with more material. But if we start with more material, then we end up with more of this stuff, which is overwhelming the signal in the first place. And so you end up back where you started. So if you can't get more of the sample to do that increase in sensitivity, what do you do then? Like if you take a few mils of plasma out of a person and you can't detect it in there, are you going to start taking 100 mils of plasma out of a person or a liter of plasma out of a person? That actually starts to become quite silly. And we'll talk about that further in a couple of slides. The other problem is the sheer chemical diversity and heterogeneity of proteins and the amino acids that they're made of. So proteins, if you hadn't realized, are made of 20 different amino acids. Some of these are non-polar, some of them are polar, some of them are charged, they're acidic or they're basic. And then we connect them together into a chain of amino acids, and the order of the amino acids then determines the way that the protein folds up and interacts with each other. And if you take the same composition of amino acids and shuffle them into a different order, even though the composition is the same, the activity of the protein will be completely different. These things fold up into different structures like alpha helices and beta sheets, so you end up with these primary structures that can then associate into protein complexes like this hemoglobin molecule, or into far more complicated structures like this proteasome. So it becomes technically challenging to deal with things that are so chemically diverse. Now, with anything, with DNA, with RNA, with any molecule, nearly all of the assays that we do these on has to be performed in liquid. We need molecules to be able to move around, to interact with each other, to be able to separate and detect them. So it becomes an issue of, if you can't solubilize it, if you can't keep it in solution, you're not able to analyze it. That then extends to the logic is, if you can't get it out of the sample in the first place, you can't analyze it. Mammalian cells are relatively weak and they're easy to break open, but bacterial cells are really tough. Now, what this diagram is showing, and the point that I'm trying to make with it, is what's written here from the article, from the quotation of the article, is that this looks very, very complicated, but it's actually omitting quite a number of the things that should be here, which means that even though this is looking complex, it's not as complex as it could be. And so what we should be thinking about is that these membranes and these cells are enormously heavily packed, full of protein molecules, full of RNA molecules, with loads and loads of stuff going on. What we want to do in proteomics is we want to get these things out and collect together all of the molecules of the same thing to increase the sensitivity of detection. So we have to overcome the chemical diversity of the amino acids. Once we remove the proteins out of their environment, they tend to precipitate, they tend to fall out of solution because they're unhappy, for want of a better term, not to put anthropomorphic personifications onto things that are not, that don't think. You need chaotropes and surfactants to help keep these things in solution, but these can interfere with downstream processing. And you need to break these cells open as well to get to the proteins and the complexes that you want. Now, there's loads of different ways that you can break cells open. You can use homogenizers, or you can use French presses, which force things through a small hole. You can use ultrasonic sound to break things open. You can use detergents to try and get these molecules and organelles free to be able to extract the protein out of them. So, in a summary, what we're trying to do in proteomics is, firstly, smash the cell open so that we can release everything. Then we want to divide the proteins up into manageable groups, where each one of these groups would be all of the molecules of exactly the same proteoform, to collect them into the same piece of space to increase the sensitivity. Once we've collected the proteins together, we actually cut them up into peptides a lot of the time to be able to put them into the mass spectrometer and measure their masses and their fragment masses, to be able to then search databases to find out what those proteins are. And then lastly, and the most difficult problem of all, is what the heck does all of this mean biologically? So because this is analytical biochemistry, I'm going to take you through how we actually do these analyses. So what we want to do, the aim of what we want to do is, firstly, identify what proteoforms are in our sample, and then quantify the amount of each particular proteoform in the sample. So here's our so what we want to do is say, okay, there's a car, there's this different car, there's this. These are all identifying the different proteoforms that are in there. But then we want to know, is there one of this car, or is there many of this car? And under different conditions of the cell, how many different molecules of the same proteoform are they? And are they increasing or decreasing in abundance? So now, to move away from my Lego to make things a little bit more simple, what we try and do is we have these mixtures of different proteoforms where we have different amounts of the different proteoforms. Sometimes we have post-translationally modified versions of the proteoforms, but all of these are together in the cell. And what we want to do is we disrupt it, we collect it together in a tube or in the bucket, and then we want to separate them back out. So they're all mixed together. We want to get all of the molecules of the same proteoform separate, into a separate bucket or into a separate volume of liquid within space. Now, ideally, we want to be able to resolve these to single proteoforms. And one of the ways of doing this is this technique called two-dimensional gel electrophoresis. We talked about this in the electrophoresis lecture. If you haven't seen that, go back and watch it, where we take the bucket of solubilized proteoforms, we put them into a pH gradient, we focus them to where their charge is neutral, and they stop moving in the electric field. We then put them on top of an SDS page gel and we separate them out by size. What we can now do is we can now cut this particular collection of proteoforms out of the gel and identify what the open reading frame was. So if we have these two proteoforms, that they're differing by this post-translational modification, but so they form two different spots on the 2D gel at relatively the same mass, but at different isoelectric points, which means that there is something that's been done to this proteoform to change it into a different proteoform. If we cut each of these out of here and digest them and put them into the mass spectrometer, we will end up identifying the same open reading frame. We may not identify what the modification is, but we know that they have to be modified. They have to be different because they're resolving to different spots. This is going to become important because this contrasts significantly to just doing peptide-centric shotgun liquid chromatography mass spectrometry-based proteomics. So an example of this is this one here. This is actually Staphylococcus, where this is one gel out of a collection of 30-odd gels, I believe. And the numbers refer to, these have been cut out and identified because these spots were deemed to be changing in abundance. So if you look at this area here, which is blown up down the bottom, in the untreated Staphylococcus, each of these spots has a certain abundance that is reflected by the staining density. When you treat them with this active honey, which starts to kill the bacteria, you can see that the intensity of these proteins disappears. So what you can now do is cut the spots out of here where they're more intense, put them into the mass spectrometer and identify what those open reading frames were, and whether, because these are all in a line at the same mass, are they just proteoform variations of the same open reading frame, of the same gene. So just to reinforce this in this very this example, where this is seagrass grown under either high light conditions, high amounts of light, rather than the control, where when you grow them in high amounts of light, this proteoform appears. It is not here at all. And so you're like, well, wow, this is a really significant change between the two samples. What is this protein? And we would have cut it out and identified it. So the density of these spots reflects the change in abundance so that you can quantify the fold change in the amount of protein that is in two different samples by measuring the density in this particular case. So how do you identify what the protein is? So in the previous example, if you look at each of these spots, you can say, okay, yep, there's a spot, but I could not tell you what those proteins are. Now, with DNA, it's relatively easy. You can either use next generation sequencing, which uses fluorescent terminators to be able to show you what a particular base is, or you can use, in this case, capillary electrophoresis, where the piece of DNA comes through, it has a reporter on it, which can then be read, and you can say, okay, well, this is the gene sequence. We talked about how dye terminator PCR works in the electrophoresis lecture, so I'm not going to go through it again. For identifying proteins, we rely on mass spectrometry to identify what our proteins are. So what we do is, in this particular case, we cut the spot out of the gel. We then in-gel digest the proteins with an enzyme to make peptides. These peptides get separated out by chromatography. A particular peptide gets selected in the mass spectrometer and creates fragments. So this is shown down in here, where we have this particular peak, which represents this peptide, which is glufibrinopeptide. And then if we select all of the molecules of this particular peptide and we smash them, we end up with this range of fragments. And we can then take that and we can turn that into a list of numbers that we can then put into database search engines. So in the database search engine, we have our acquired spectrum of the fragments of the peptide, and we have a database of protein sequences. And what we can do is we can take each of the sequences out of here. So this is the sequence for myoglobin, and we can theoretically cut this up with trypsin. So we can go through and say, okay, trypsin cuts at every lysine and arginine. We start at the beginning, we go through, here's a lysine, I'll cut there, and I'll create this particular peptide. And so this is the collection of tryptic peptides that would be made from this open reading frame that's in the database. We can calculate the mass of the peptide, which is the mass that would have been selected for fragmentation in the raw data. And then we can take each of these sequences and create a table of what the theoretical fragments would be from fragmenting that amino acid sequence. So that creates our theoretical database that we can use to search things against. And so then we have our acquired spectrum from the mass spectrometer that we've turned into a list of numbers, and we compare these things. So we say, okay, I've got a peptide at 1606 from my entire database of theoretical peptides that I've made. Give me the list of everything that is this mass, plus or minus an error. And the error is normally very, very small. It's normally about 0.02 to 0.05 of a Dalton. From that list then, compare the fragments of the things that are in the theoretical database to the fragments that's in my data. And the more fragments that match, the higher the score that you get. You can then... You can then start matching these numbers together. So 175.1 in our raw data matches 175.1 in our theoretical theoretical data. 288 matches this one, and we go through and we keep matching. And the more things that we can match, the higher and higher the score gets. What we can then do is do probability-based statistics on these things to have the probability that the match to the database is not a random event. And we can also do statistics to work out what is the likelihood, the expectation value that things are real. And this is analogous to a p-value. So the smaller this number, the more likely it is to be correct. The score does depend on the size of the input data from the mass spectrometer and also the size of the database. So statistics will change depending on what goes in and what comes out. Now, we'll go back to talk a little bit about 2D page proteomics because it's important, because we've been talking a lot about proteoforms and the need to identify proteoforms. But interestingly, in modern day proteomics, we don't measure proteoforms very well. In 2D electrophoresis, we are working on and resolving intact proteoforms. We are able then to quantify the differences in the abundance before we identify. So we can run all of our gels and we can unbiasedly look at the density of particular spots across the treatments and go, those things are the interesting things. There is a X-fold difference between untreated and this, and I want to know what that protein is. So then I only have to run, in this case, 18 spots through the mass spectrometer, massively cutting down on my instrument time. However, it requires a lot of protein. So to run triplicate gels, you really need about 1 milligram of starting material. You're looking at running between 100 and 300 micrograms of protein per gel. And so that translates into needing about 400 million cells to be able to do this. Now, that's not too difficult from cell culture, but it depends on what your samples are. You typically only see about 1,000 proteoforms on a gel, and some of those spots, some of those proteoforms are modifications of the same open reading frame. So you're probably only seeing 500 to 600 open reading frames, but you're resolving the proteoforms out. So the reason that you don't see more is because of that dynamic range problem that I talked about in the beginning, where the concentration of the highest abundance to the lowest abundance. So in these areas here, there may be proteoforms in here, but because you can't see them, we can't quantify them. So we don't know whether or not there are things in here and whether or not they're changing in abundance. There are strategies that we can use with fractionation to be able to see deeper and deeper, but by fractionating, we need more material. The most used alternative to this is what's called shotgun mass spec. And so what we do is we take the entire proteome and we turn it into peptides, typically using trypsin. And so peptides are more soluble. We can fractionate them at higher resolution, so we can separate them out from each other more easily. And they're simpler to analyze by the mass spectrometer. They ionize far more effectively, and you get more sensitivity with peptides than you do with intact proteins. This is typically interfaced with reverse phase chromatography, which is to reduce the complexity of the sample going in. So it separates the peptides out from each other in time and staggers them going into the mass spectrometer so that it gives the mass spectrometer time to fragment each peptide as effectively as possible to increase the size of the data. The alternative is to try and measure things by top-down mass spectrometry. And what you find with back in the mass spectrometry lectures, we talked about electrospray and how that creates multiply charged ions, and we showed how proteins create these multiple charge spectra, and so the signal is actually diluted out across different charge states. But what we can do is we can run the same sample across, and we can measure, work out what the mass of the intact proteoform is from this charge series, and then fragment it to get information that will tell us what the open reading frame was. So in these three cases, the open reading frame that has been identified is exactly the same. But the differences in the measured intact mass, where we've gone from a mass particular here to something that's 14 Daltons larger, is because this arginine is being acetylated. It's having a methyl group put on it compared to this proteoform, which increases its mass by 14 Daltons, and we can measure that. This proteoform has also had a phosphorylation put on it that increases its mass by 80 Daltons. So by mapping to the same open reading frame, but again having different intact masses measured, we can say that they are different proteoforms. So we can also measure the abundance by the height of these charge states. Again, we get low proteome coverage in a similar manner to 2D page because the signal is spread out across numerous different charge states. And so what typically needs to happen is extensive fractionation. In shotgun peptide proteomics, you can take a complex mixture and just inject it straight into the mass spectrometer. You can't do that in top-down. So for one single sample, it can take days of mass spectrometry time, which is not practical. Whereas peptide-centric proteomics, we can identify the products of up to 10,000 open reading frames in a single sample in 3 to 4 hours. And as I said, the majority of proteomics projects nowadays are done this way. So the problem with doing shotgun proteomics is that we have, so we take our proteoforms and we digest them with trypsin, and if we have two proteoforms that differ by PTM, they will create the same peptides, except for this one that's modified. So this now relies on the fact that to be able to distinguish, once these are mixed together, which is what typically happens, you take a cell digest and you digest it all together, you don't separate the proteoforms out beforehand. it relies on the detection of the unmodified peptide and the modified peptide to say that there is a PTM in here. The other thing is, I've colored these things to make it easier to differentiate, and colored the peptides here to make it easier to differentiate, but excuse me, but in reality you don't have them labeled in that way. They're all a grey bucket that needs to go into the mass spectrometer. So how do you know which peptide the proteoform came from? And if you don't detect the peptide that defined the proteoform, you can't actually distinguish the different proteoforms are there. Now, we can reduce the complexity and try and resolve this issue a little bit by doing fractionation. So we spend, and many people spend, a lot of time optimizing fractionation to try and increase the data that they get. Now, this is a slice out of a 2D gel. You've seen this in the electrophoresis lecture, where I've said that all of these proteoforms are actually in this block of the gel here. So what we can do to reduce complexity is separate out the proteoforms by size, and then in gel digest a block of it, and then separate those out by chromatography and into the mass spectrometer. And so then what we can do is say, in this particular case, what we would get is, we would get a collection of matches to an open reading frame. In this case, we would be getting matches to an open reading frame that is 200 kilodaltons. And so if this was not undergoing proteolytic cleavage, it'd be up here, but it's not. We're detecting it in a fraction that's between 55 and 43 kilodaltons. So we then realize that there's got to be some kind of proteolytic processing that's going on of that open reading frame. So we've already given more information. We haven't solved the mystery, but we're getting more information about what the heck is going on. So unless we identify the peptide that defines the isoform or the proteoform, we can only identify the ORF. If it's post-translationally modified, but rather than an amino acid substitution, it's unlikely that things are going to get found. So in this particular case, this demonstrates the difference between an amino acid substitution, where it starts to fundamentally change the mass of not only the peptide, but also the fragments that we are going to find. This relies on the mutant sequence being in the database. If it's not in the database, we won't match it. And so if the modifications change these masses. So, as I said, if the peptide defining the proteoform is not detected, we don't realize that the thing is actually there. So this is a real example. This actually comes from a 2D gel spot, I believe. But we've got two options here where we have the same group of peptides matching to two different proteins, and the only difference between these two sequences is this D to E mutation in a peptide that has not been detected. So we can't actually say whether or not the change in abundance is due to a proteoform change or something else. So just to summarize a whole lot of stuff that I've been talking about, the reason that you would typically work on peptides is because they're very, very soluble, they're easy to resolve at high resolution in chromatography, and it's easier to detect and get good fragmentation data from them in the mass spectrometer. We can get high proteome coverage, but lower individual protein or proteoform coverage. The majority of the proteins are identified by only 1 to 2 peptides per protein. And we often lose this isoform information. So why would you do it? Because you get a heck of a lot of information in a very short period of time, and you can use it as a starting point to start to answer your question and then resolve those questions by other means. Proteins, on the other hand, working at the proteoform level, we can differentiate different proteoforms and we can fractionate them before we identify them. We can get greater individual sequence coverage to try and figure out whether the proteoforms are due to amino acid substitutions or whether they're due to post-translational modifications. And in addition, we can keep complexes and protein interactions together to better isolate and understand what the heck is going on. Those were all identification problems, and as you've probably realized, identification of the number of proteoforms that is in there are very important, and the identification of the presence of modified proteoforms is really important. But it is also the amount of a particular protein or proteoform present that affects the phenotype. So the ultimate objective of proteomics is actually quantifying the changes in protein abundance under different conditions. Temperature stress in bacteria. What happens to normal cells in cancer or infection? What happens to a cell during pathogen invasion? Do you get changes from phosphorylation? Do you get more proteoforms being phosphorylated or dephosphorylated? In 2D page, as I said, we do this by measuring the abundance of particular proteoforms. So you can see the density of this spot has changed between this sample and this sample. These spots are changing in abundance. These things can be very, very subtle. But now what I can do is I can cut out the individual ones and identify what they are. With shotgun and even taking gel spots and digesting them, we quantify the abundance of open reading frames rather than proteins. And so we're inferring the change in abundance of an open reading frame through the quantification of the amount of a peptide. So we can do this by label-free methods where we prepare the sample separately, we run them separately through the mass spectrometer, and we compare the area under the curve for the same peptide in these different runs. It relies on the reproducibility of the mass spectrometer, and you typically have to run quite a number of technical replicates. In this case, we're having to identify what a feature is, what a mass in the chromatogram is, before we quantify it. Again, we're identifying open reading frames rather than proteoforms. This then leads to a thing that's been going on for 25 years called the protein inference problem. If you have multiple proteoforms, but they share the same peptides, how do you assign the abundance of a particular peptide to a particular proteoform? And that's really, really hard. One of the other ways that you can measure and quantify stuff is by doing labeling experiments, where you can grow different cells in culture with different metabolic labels. We talked about this a little bit in the mass spectrometry lecture, so I'm not going to go through it again. These things allow a reduction in the number of technical replicates that get done, and therefore reduce the amount of instrument time that you need for analysis. So this leads to the question, which way is actually better? And the answer is, how long is a piece of string, which is twice the distance from the middle to the end. So with 2D page, you can keep your proteoforms intact. You can do quantification before identification. The spot defines the proteoform. There is a lot of hands-on labor that goes in 2D. It's not that bad. The literature makes it out to be a horror show, but it's actually not that bad. And you can parallelize a lot of the analysis that you can't do that in mass spectrometry. You typically get higher sequence coverage of individual proteins and proteoforms, but you may get less sequence coverage unless you start to do significant fractionation. In shotgun mass spec, you're analyzing peptides and inferring proteoforms or the products of open reading frames. you can lose proteoform information if you don't detect the peptides that define the particular proteoform. You have to do identification before you do quantification. And so to be able to quantify a particular isoform, you need to be able to detect that peptide. You do get higher overall proteome coverage at the expense of individual protein or open reading frame sequence coverage. But the other biggest advantage that people love is that you get automated constant running, where you can load 20, 30, 40 samples into the autosampler of the mass spectrometer and let it do its thing over how many days it needs to do it and go off and do other things. Everything that we've been talking about up to this point has been about individual proteins and individual proteoforms, which is the way that the majority of quantitative proteomics works, where you're looking at the change of abundance of individual proteins and proteoforms. That's not how biology works. They don't act in isolation. They interact with other proteins and other complexes. So you can see examples of this here, where you've got two different proteins and they form a complex like cytochrome C. You've got different proteins changing their conformation when they interact with each other from what they were. You've got proteins interacting with DNA or with metabolites or lipids, where that the interaction between the two things changes its form or can lead to the formation of protein complexes, where you need particular cofactors to be able to assemble complexes. So ideally, we want to measure and quantify these proteins and these protein complexes. This is actually really, really difficult because the methods that we've been talking about, to be able to keep the proteins and the proteoforms in solution, we use some fairly harsh conditions to keep them in solution. And so we can't do that because we'll disrupt the interaction between these different proteins. So there's two main ways that people get around this. The first one is this thing called chemical cross-linking, where you use a cross-linker to bind complexes together. This kind of works, and you do get, but you can get non-specific. You can get things that don't bind to other proteins. You can get cross-linking of things that shouldn't necessarily be cross-linked. There's got to be a lot of validation that occurs within these experiments. The other way of doing it is actually using milder conditions to try and stop the complexes from becoming insoluble. So one of the main methods that people do is this idea of separating by size exclusion chromatography and then doing a digestion of the particular fraction. So you can see the number across the bottom here is the fraction number of the separation by size, and the molecular weight of the fraction is up the top here. So what we've got here is each one of these lines is a different detected proteoform within this signal zone. So these are all, so the individual sizes of these complexes is only about 20 to 25 kilodaltons as shown down here. But this particular complex is eluting out of the size exclusion column at 338 kilodaltons. And then we take the fraction, we turn it all into peptides, we do shotgun MS, and we find peptide evidence for each one of these things. So this complex is made up of each one of these things. And you can do this under different conditions where each one of these lines is a different set of conditions. And you can look at the abundance or the intensity of the complex and work out the differences in the amount of complex between different treatments. And you can see that what they've done then is they've modeled the way that the complex actually assembles, where you've got, in some cases, you've got the assembly of just this bit and not a lot of this part by itself. this part to exist needs to be complex to this. You don't see this part of the complex by itself in the size exclusion chromatography. The complementary technique to this is cross-linking, where you take a cross-linker and you cross-link lysine residues across the protein. Some of these links will be within a proteoform, some of them will be across different proteoforms. And then you digest these with the links intact. You run them through mass spectrometry and then through specific data analysis software to be able to say what peptide was linked to what peptide, and be able to start generating interaction maps and models, because the interaction map comes from a particular peptide from one proteoform being detected joined to a different peptide from another proteoform. But then when you get ones that are intra-linked, where you're linking together peptides from the same proteoform, it can start telling you what residues, what amino acids were close to each other within a proteoform to be able to enhance that modeling. Once we do that, we can actually start drawing these interaction maps. This is an example from string, where a collection of proteins all belonging to the photosystem, it shows that these proteins interact and how they interact and how that information is obtained. And so you can draw these big interaction maps with these little boxes beside them showing you the changes in abundance in particular treatments. So this particular protein you can see is going up in abundance in a certain treatment and a little bit less in a different treatment. And that protein has been found to interact with this protein hub, which has these things complexed with it, and they're varying in these amounts. So this particular hub is this particular protein complex, and you can see the differences in protein abundance of the components of the complex. Before we finish, I'm going to take a quick detour into system-wide analyses, just to give you an idea that, and this works in a similar way to proteomics, where we're measuring the change in abundance of different molecules under different conditions to add another layer of what is going on within the cell. So in this particular case, we're showing examples of all of the different classes of lipids that they are. Now, the problem with lipidomics is that you get a lot of isobaric compounds that have different structures but exactly the same mass, and it's very, very difficult to be able to determine what is what. And so we can use lipidomics to look at different things that happen under different conditions. So in this particular case, we've got human breast milk and the diversity of different types of phospholipids within human breast milk, and then a different stage of human breast milk. And you can compare this to the levels of different things in different infant formulas. And so what you could do is start to look at this and go, well, wait a minute, these infant formulas, they actually have a different composition of lipids compared to what human and mature milk is. Maybe we need to modify, if we have to use infant formula, maybe we need to further modify the composition of these things to be able to create a product that is more similar to what it should be. This is another example where this is looking at the differences, the change in lipids with coffee consumption compared to the control. And you can see that you actually get changes in plasma lipids in response to coffee and caffeine. So their findings suggest that coffee alters glycerophospholipid metabolism and may provide insights into the mechanisms by which coffee may exert its health effects. If you want to actually find out what those health effects were, go and hunt down the paper. Metabolomics and lipidomics, they use similar technology to proteomics. except we don't typically use electrophoresis. We typically use just chromatography and mass spectrometry, where we generate, we individually run our samples. We generate these tables where we have the intensity, where we identify particular metabolites and the intensity of those metabolites. We can do principal component analyses to look at controls and treatments, look at things that are changing in abundance and be able to identify them and do that quantification to try and figure out, okay, can we come up with some kind of a diagnostic? So in this particular case, and I'm sorry, I can't recall what the examples are, but what this is showing is the amount of particular amino acids in a particular patient, what you can measure. All of this now we can link together. So we can bring together transcriptomics, proteomics, the interactome, phenomics, lipidomics, metabolomics, integrate all of that data together to start to do modeling to try and figure out what's going on when things change at this level, or when the genome starts expressing different things. And this is all under the field of systems biology. So with that, hopefully what I've shown you is that what proteomics aims to achieve is to characterize what proteins are being produced at a certain point in time in a certain cell, how much of them are being produced, and how are they being modified. And this is one step in the global analysis of all of the biomolecules within a cell to try and understand what's going on in disease or infection when these molecules change. Can we come up with biomarkers, and can we come up with therapies by understanding how the phenotype, how individual parts of the phenotype change over time?